our moderator for this lecture. Thank you very much, Dr. Arya.
Thank you. Thank you very much, Dr. Jeffy. I would like to again welcome everyone to our wonderful webinar today. If I could just share my screen. All right, so again, thank you very much for participating in the Innovations in Aging Science, Animal Models for Aging from Simple Organism to Vertebrate. We are really thankful we are able to get a lot of science and a lot of knowledge from Dr. Rika, Pak Firzan, and Dr. Abe, and Mr. Semi. So today, we would like to begin, as Dr. Jeffy said, by stages. Let me introduce our first presenter today, Dr. Rika Indriastuti, graduated from Nara Institute of Science and Technology with many reputable involvements in microbiology, such as as the technical manager of microbiology analysis in Bogor Agricultural University, and even an editor of a journal for Sumber Daya Hayati. So we are looking forward to hear the enlightenment from Dr. Rika on how we can utilize yeast in this aging study. Please, Dr. Rika, you have 20 minutes.
Thank you, Dr. Ariel, for introducing me. That was a really nice introduction. So once again, everyone, my name is Rika Indriastuti. May I be enabled to share screen?
Yes, please.
Thank you.
Okay.
Can everyone see my screen?
Perfectly.
Yes.
Okay, thank you, sir. Then let me start. Assalamualaikum warahmatullahi wabarakatuh. A very good afternoon for everyone. And thank you for the committee who invited me today. I am very happy to be here. So my name is Rika Indriastuti from IPB University. And I work in the Laboratory of Microbiology, Department of Biology, IPB University. And my team and I currently work quite intensively at yeast, and we use yeast as a model, especially to understand cellular aging. So when we talk about, does my slide change?
Yes.
Okay, so I need to confirm that, because usually during my lecture, there's some problem that my slide couldn't change to the next slide. So model organism is somehow a very powerful tool to understand a very complex, especially how cells work. In terms of aging, aging is also a very complex phenomenon, and using model organism makes the analysis somewhat simpler. But anyway, the cell works in terms of aging is obviously complex, so that researchers try to reduce the analysis, either reducing the focus of analysis, such as, they will just focus on a DNA repair system, or in the DNA replication system, or on the other hand, we can use a model organism to understand that complex phenomenon such as aging. On the left figure, you can see various model organisms that has been used widely by researchers. We can see that there is an E. coli. This is a prokaryotic microorganisms, and here Saccharomyces cerevisiae belongs in the yeast. This is also the microorganism that we use in the laboratory to study aging. This is a eukaryotic and unicellular microorganisms, and later on we also know some other eukaryotic organism which more complex than E. coli or Saccharomyces cerevisiae, such as Drosophila or C. elegans, and including mice, as Dr. Jeffrey mentioned before. So in terms of yeast, there are two yeast species that widely used to study numbers of physiological or cellular phenomena, including aging. They are Saccharomyces cerevisiae, and Saccharomyces pombe. The main difference between these two species are Saccharomyces cerevisiae is a budding yeast. You can see here in the picture, there's a bud come out of the mother cell. So this is a so much smaller daughter cells than the mother cells. But the Saccharomyces pombe, this guy is not a budding yeast. This is a fission yeast. So they will have a same size of a daughter cells to that of mother cells when they divide. So they will do binary fission, just like a mammalian cell. So these two are used nowadays to study cellular phenomena, including aging. You can see here numbers of study, such as aging as well, regulation of gene expression, signal transduction, cell cycle, apoptosis, even neurodegenerative disorder can be studied using this system. And among the advantages, or also some of the disadvantages of using yeast as a model organism, we can see here that 3 points somehow making it a relevant or sophisticated system to study aging. Here, nearly about 1,000 yeast gene, or approximately 17% of its genomic background, is a member of orthodox gene families associated with the human disease, and that some of the genes that involve in aging somehow a complement between yeast and the human system, so making it more relevant or support the relevancy of using yeast as model organisms. And this Saccharomyces cerevisiae and Saccharomyces pombe has been fully sequenced. I mean, the DNA has been fully sequenced, and all this data are available publicly. So we can access the data very easily. And another advantages for us, especially we work with yeast, and we found that these Saccharomyces cerevisiae and Saccharomyces pombe, they have a gene deletion library that are available in public. This is commercially available, so we can purchase them and we can use them, especially to study a functional gene analysis. This is a very, one of the advantages of using this, because the gene deletion library is quite a databases that makes us available, or that enable us to study a number of cellular activities, including aging. And this is one of the manufacturer that produced the gene deletion library, the Pioneer from the US, and we use this in our laboratory. And each of the non-essential genes is replaced with the antibiotic-resistant genes. This is NMX4, and those non-essential genes will be replaced by homologous recombination. And you can see that are numbers of primers that can be used to detect the exact place of this replacement of antibiotic-resistant modules in each of the essential gene yeast here in Saccharomyces pombe. And the technical advantages. When we use microbes, we understand that they have a very short lifespan. Like yeast has only 2.5 hours to double. So it's very easy to manage. It's very easy to maintain in the laboratory. We understand how to grow them. We understand how to manipulate them, physiological way or genetic way. So toolkit to manipulate or do some genetic engineering of yeast is available. So that's also some of the advantages of using yeast as a model to understand a complex phenomenon. When we talk about aging, aging is, once again, is obviously a complex phenomenon. Here, Brodsky and colleague make a review about they divide aging into two terms, primary aging and secondary aging. You can see here in the primary aging, it's basically how cells naturally age, because the production of ROS or reactive oxygen species, and a secondary aging is we can say an environmental influence, such as a food intake, or here you see a couch, meaning that some people may do exercise and some people may not do exercise. It's also influence the characteristic of secondary aging. And on top of that, you can see that many of the processes actually involved in the generation of aging. In terms of cellular aging, we understand that the aging can be happen or can be occurred in the individual cells. You can see here, this is a review by Strew et al. 2017. They managed to figuring the differences between young cells toward that old cells. Basically, in the young cells, you can see everything works really good. When there is a mitochondrial activity, they will basically produce ATP, produce energy, and a very minimum accumulation of free radicals or reactive oxygen species compared to that the old. And when there is a misfolded protein or unfunctional protein, young cells will be easy to make a quality control to that. They will use a chaperone's autophagy proteasome to, diminish this misfolded protein and make them even as a source for energy by autophagy. But instead, in a such sophisticated mechanisms may occur, but in a slowly manner, or even somehow it couldn't be occurred because the condition is very severe. As you can see here, that's many stresses occur. Here you can see the stress come from the reactive oxygen species, imply with the oxidative stress conditions, and the endoplasmic reticulum stresses is somehow also imply with the proteotoxic stress due to the accumulation of unfunctional protein. So such aged cells, old cells, cannot undergo a sophisticated system as a young cell, so that it will lead to the lower cell viability and also the cell death. So when we talk about aging, or contrarily longevity, the pathway that occurs in yeast to the other organisms or model organisms here, like C. elegans and mice, is somehow conserved. Well, this is quite a simplified pathway. Once again, aging or longevity or increase of lifespan has a very complex system. And but you can see here that numbers of key regulatory systems, such as mitochondria, or here a protein called SIR2, silent information regulation, is involved in this longevity pathway. Whereas when the cells are influenced or treated in a calorie restriction environment, so a mitochondrial activity will increase, and that will somehow induce the activity of SIR2 protein and some increased life. Such pathway also occurs in a C. elegans and in mice. Along with the more complex systems, so the more complex the pathways are. And the human system also has a quite similar pathway, but it is very difficult to find out the lifespan of a human. So there are some of the physiological changes that observed to conclude whether the cells have a prolonged lifespan or not. But this picture represents that there is a conserved pathway in between model organisms to that what happened actually in a higher integrative system, especially in not only longevity pathway, these also have a quite homolog conserved pathway in terms of another physiological phenomenon. here, or molecular phenomena, as you can see here. So we use yeast as a model to understand aging, especially in its signal transduction pathway. This is one of the one of the key assay that we use when we would like to study aging in yeast. So since yeast is a microorganism, and when we grow microbes in a culture, in the laboratory, in a batch culture, like in a medium, in an allomer, let's say, we understand that these microbes will go different phases throughout their growth. They will undergo a lag phase, log phase, or exponential phase, stationary phase, and the death phase. This is also one of the advantages when we use microbes, because when they go a log phase or exponential phase, we understand that they will double their cells. And when they go stationary phase, they will stay as long as they can to stay viable prior death. Because of that, when we use the microbes in terms of yeast, we can measure both a replicative age and a chronological age. Replicative age means that this is a mother cells, and each of mother cells will make a bud, which is a daughter cells. Here, the smaller size of the cells is a daughter cells. And each of the mother cells can make a series of daughter cells, and we can count how many daughter cells they will make throughout the specific period that you treated. As you can see here, the number of the generation, or the number of the daughter cells, represented by the number of the bud scars in each of the mother cells, represent the replicative age, and we measure this in the log phase. On the other hand, we can also measure the chronological age. We will focus on the stationary phase, where the cells is no longer doubling, no longer undergo a log phase, but stay on the stationary phase. And on that phase, we will count how long they will stay viable. Will it be like 10 days, 15 days, or 20 days? So we can measure how long they stay viable in the stationary phase. So that means a chronological age. So by using yeast, we can measure both replicative age and that the chronological age. So one of our focus is nitric oxide. So this is one of the signaling molecules that is very important in human system. They involve in three very important system: neuron system, immune system, and cardiovascular system. It is understood that along with the aged human, the production of nitric oxide in the human system is reduced. So some of the study revealed that how involved in the development of some of the neurodegenerative disease or degenerative disease. So we would like to understand what this molecule do in the cells, because this is a very bizarre molecules. You see, this is a radical molecules because there is an extra electron that is unpaired. So this belongs to the radical molecules, but this is a very important molecule. So we use a cytosaccharomyces poly to elucidate what the function of this nitric oxide in terms of cell survival / aging. So we use a genomic study, functional gene analysis throughout the genomic of the cytosaccharomyces poly, and after long searching, we found a number 2 genes actually involved in the modulation of nitric oxide. Basically, we measure the intracellular nitric oxide in numbers of deletion cells, gene deletion cells. This is a number. This is the genes that we expected to have activity in modulate nitric oxide. Basically, when we delete two genes, this SPAC and FMD2, we found an increase in the nitric oxide both in log phase and stationary phase. And interestingly, when we interfere the cells by adding an exogenous nitric oxide, well, exogenous nitric oxide somehow increase cell viability. But in double deletion mutant, such an increase in the cell viability cannot be occurred. So it means that these two genes involved in the modulation of nitric oxide that exert to the cell's viability. And we did a transcriptomic analysis, and we found that numbers of gene clusters that is modulated by nitric oxide. It's quite complicated data, but please just focus on the biological process. Here, genes that involve in iron assimilation or iron homeostasis system is upregulated, but those genes involved in a respiratory chain reaction here, cellular respiration or generation of ATP is downregulated. So this is quite interesting phenomenon. So we managed to do a functional gene analysis, a gene deletion study, and etc., and come up with this pathway of nitric oxide. I'm sorry to making it fast in the explanation, but we did numbers of study here. Here, the nitric oxide as a signaling molecule will induce the transcriptional level of those enzymes that involve in the detoxification of oxidant. Here we use H2O2 as a source of oxidant that in the cells will be converted into radical molecules. So we managed to measure the activity of each gene that was suggested to be regulated by nitric oxide. And on the other hand, you can, if you remember that nitric oxide repressed the activity of mitochondria so that it may reduce the production or the accumulation of a reactive oxygen species by mitochondria. And if you remember that the nitric oxide also increase the cluster of genes that involve iron homeostasis. So we suggest that they increase the transcriptional level of some of the iron transporter protein, iron transporter genes, and that's because the intracellular level of iron in the cells is very depleted or very low. And such a low concentration of iron is good for the cells, because when there is iron in the cell, H2O2 can be converted into another strong radicals for the hydroxyls because of the reaction with iron. And the hydroxyl here is somehow more stronger radicals. It can directly bind to the DNA. So such regulatory activity by nitric oxide may reduce the accumulation of reactive oxygen species, thus resulting in a cell survival / aging. We did both in a log and a stationary phase. So this is also one of the study that we've done in this one in yeast Schizosaccharomyces pombe, but this one in yeast Saccharomyces cerevisiae. We try to expose the cells with environmental stresses, which is one of the trigger for aging. Here we use a high temperature, which in turn in cells will be, somehow causing accumulation of the reactive oxygen species.
Maaf dok, waktunya udah habis. Mohon maaf.
Yeah.
Ya, silakan.
Thank you, Padu. And that intracellularly, nitric oxide is synthesized by the amino acid arginine by this protein called TH18, and that the nitric oxide will further induce the transcriptional factor called MAC1, and that the induction of MAC1 will result to the induction of the transcription of its downstream gene. One of them is CTR1. CTR1 encodes those protein in the membrane that involve in the import of copper. And we understand that copper is the cofactor of SOD, superoxide dismutase, one of the enzymes that can tackle or inhibit or diminish the activity of reactive oxygen species. And this system may result in the capability of these yeast cells to stay viable. And that's also one of the factors to prolong lifespan. And that's one of the story that we studied about the signal transduction of cells in aging, especially involving nitric oxide. And this is one of the other study that the application of yeast as a tool, as the tool for study aging that we did in our laboratory. So this is a review. I think this is a very sophisticated figure by Zimmerman et al. Back in 2018. So when we have a compound library, many natural products from plants or bacteria or any natural products, we can assay the bioactivity by using this. You can see here, there's a possibility that the compound may not be able to even go into the cell, imported to the cells, or probably some of them can be imported to the cells, but then exported again, or there's just no activity. That's possible. But maybe some of the compound can extend the lifespan of yeast by simple assay that I explained before about the lifespan. So when it is have an activity to extend the lifespan on yeast, we can further analyze in the multicellular organisms. And when it still has the activity to induce or to prolong its lifespan, it's a potential anti-aging compounds. When it's not, it still have a biotechnological application, because yeast is a fermentation agent, also a host for production of some of the protein heterologous, especially human recombinant protein. And when probably some of the compound may toxic to the yeast, causes death to the yeast, that's fine, because when we further study them in the human cells, when it's still toxic, we can use, or we can further study as an anti-cancer compounds. When it's not toxic to the human cell, there's a possibility that the compound actually have an anti-fungi activity. So this is
Mohon maaf, Dok. Waktunya sudah lewat, Dok.
Okay. So this is our study by using yeast as a model organism. I cannot explain one by one, but as Dr. Ariel say, my time is up, so probably I will close my talk. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh. Back to you, Dr. Ariel.
Thank you. Thank you very much, Dr. Rika. As we said, if there are any questions, please do write it down in the chat, or we can always ask it directly later in our Q&A session. one thing I learned is never to underestimate yeast, because even I think the most recent paper in Nature talking about how synonymous mutations is actually dangerous is actually done on yeast. I think it came
Exactly.
out. I think it came out this week or last week. I forgot, but I was just reading about it. So that was fantastic. It was a fantastic paper. All right. So thank you very much, Dr. Rika. Now
You're
let's
welcome. Thank you.
thank you. Okay, now let's move to our next presenter, Dr. Firzan. Let me show his reputation. Having earned his PhD on pharmaceutical sciences, a graduate School of Medical Sciences from Kanazawa University, Japan, and a Master of Biomedical Sciences in Microbiology and Immunology from Australia. We are very excited to hear from him any perspective in using Fly, especially as a founder of Fly Indonesia and Hunas Fly Research Group. So I'm sure he has a lot of perspective to share with us on how to use Drosophila for the investment of science. Please, Dr. Firzan, you have 20 minutes.
Okay, thank you very much. I will start this screen sharing. Is it okay now?
Yes, but it's not full screen.
Yeah. All right.
Now it is. All right.
Okay. Thank you very much, Dr. Ariel Pradipta. I will start my presentation. Today, I will talk about the use of fruit fly as a versatile model for aging study. My name is Firzan Nainu, as introduced by Dr. Ariel. I work in Faculty of Pharmacy, Hasanuddin University, Makassar, Indonesia. And the outline is, I will first introduce what is Drosophila, because maybe not so many people use Drosophila, especially in Faculty of Medicine or Faculty of Pharmacy, or the ones presented in here maybe will trigger your, will encourage you to use it. And I will present what has been known about aging science using Drosophila melanogaster, and what we are doing at my facility in Hasanuddin University. First, I would like to show you the picture of Drosophila. So this is actually an insect, an invertebrate, and we can see in the left panel at the way up, this is a male. Sorry, I will use the pointer. Just wait a second. Okay, this is a male Drosophila, and this is a female Drosophila. And then the chromosomes are four chromosomes are in Drosophila compared to humans that are 23 pairs of chromosomes. They only have four pairs. And as we can see in here, we can see that male Drosophila has, we can distinguish male from females. Basically, males are smaller. This is maybe similar to humans. Sometimes women are bigger than males. Sorry, humans in female are bigger than male. This is also true in Drosophila. So that's why when one male is inside the file or inside the cage with one female, sometimes the male died, unfortunately. So one male in here, we can see the black at the posterior, we can see the black things on the abdomen part. So we have head, thorax, and abdomen. And we can see in the really back, we can take a look at the really black strip of the male part. And they have this, what we call genital claspers. And this is particularly when we see usually we use microscope to see Drosophila. We use stereo microscope, and we can take a look at different stages. This is, for example, particular virgin female that just eclosed from the pupa. Okay. To make it easier for everyone, actually, I prepared one article in Indonesian journal just to make it easier to understand what is the features of Drosophila and what are the advantages using Drosophila in biomedical research, particularly using in drug screening. And you may take a look at this paper I published in 2018, and just to make it one particular notion that we can use Drosophila for forward genetics and reverse genetics easily, and we don't need ethical clearance, just like yeast. And the life cycle of Drosophila is really fast. We can have, in 10 days, we can have new adults. And as you can see in here, there are several stages. First is embryo, and then the embryo becomes larvae. Larvae become this first instar larvae, and then becomes second instar and third instar, and become pupa and becomes adult. And we kept the Drosophila in a file like this. So it is specifically constructed for Drosophila. So we call it Drosophila files. Okay, so in 10 to 12 days, we can have from one female, we can have around 50 offsprings. So it's really large. We can only have 3 files of this, and we can have hundreds of new offsprings in only one day. And what is particularly interesting is 75% of known human disease genes actually have a recognizable match in genome of fruit flies. And based on new articles, we know now that around 68% of Drosophila genes human cancer-related genes are also available in fruit flies. So if we take a look at papers presenting by Drosophila researchers, we actually know that many diseases are now being investigated using fruit flies. One of particular use is for cancer disease and also aging. And the reason why is because the same genes organize the Drosophila bodies as well as human bodies. So we know Hox genes, and it is particularly interesting because Hox genes previously discovered in fruit fly before discovered in mammals. And we now know that when we impaired or we disturb those genes, we may see defects in Drosophila as well as what happened in humans. So this is an example of Hox genes, hedgehog genes. This expression, we can see two wings, and similar things happen in the toe, in the hands of the human. And the same genes also structure our nervous system. So that's why many papers come out from Drosophila researchers also talk about in nervous system and influence our behavior. Then I just want to inform everyone that even Drosophila is really small and easy. Sometimes people say this is really maybe not interesting creatures or insect. They actually, around 10 people already got their Nobel Prize in Physiology of Medicine because of this insect. And the last time researchers got Nobel Prize in Physiology or Medicine is actually in 2017 when Hal, Rosbach, and Young about the circadian rhythm. And interestingly, Drosophila also can develop many diseases such as cancer, as I said previously, epilepsy, Parkinson, Alzheimer's, and many are written in Nature's paper or Cell's paper, as you may later found if you search about it. And then, yeah, we can also now study about jet lag genes because it was found in flies. And then also when Drosophila consume, for example, coffee or tea or something like that, they also experience they're difficult to sleep, similar to us, and also get drunk by alcohol and get addicted by alcohol as well, and many things. For future information, you may visit the link in here. And then, yeah, just as I said, in 2017, the last time when Drosophila researcher got the Nobel Prize about the discovery of molecular basis of circadian rhythms that now has been known play a role, a really significant roles in humans' life, as well as in other mammals. Okay, how we maintain Drosophila in lab? We usually put it inside incubator like this, and inside the incubators there are so many files like this, and we name it based on the genetic background of the flies. So sometimes, for example, in here Oregon R, it means that it's a wild type, and then sometimes we have mutants, transgenic flies, and everything. We put, of course, the label to know. And then we use the stereo microscope for taking a look how flies distinguish between male and female and everything based on the features that we have. And we also can inject drug into Drosophila using nano injector. The usual volume is around 50 nanoliters. So we can dissolve drugs in particular solvent, and we inject them into the flies to test the drugs. And then this is an example of mutants and transgenics of Drosophila melanogaster that has been prepared by Andres Prokop from University of Manchester. We can easily learn about flies, every type of flies using this kind of pictures, and you can download it for free. And one of particular techniques that we use in Drosophila that are so interesting is the forward and reverse genetics. I remember when Dr. Jeffilano asked about this kind of features previously. Yes, we can do it in flies, but yeah, it requires a lot of money, of course. But it's interesting. So in forward genetics, we choose random. We give a particular substance, and we will have many mutations occurred in flies, and we select the phenotype of interest. Of course, for example, in aging flies, we will take a look which fly will age faster, and then we choose that particular phenotype, and then we will examine what kind of genetics happen, what kind of things happen in the body of this fly. And then we also can use this transgenic system. We call it Galforia system. In this particular technique, we use two different flies. First is the fly that has Galfor. Galfor is transcription factor from yeast, which Dr. Rika Indri just presented. We inserted the Galfor into the back of the promoter. This particular fly will produce Galfor protein, and we do mating with the US line. The US line is the line that has gene of interest. For example, if you're interested about one particular gene that play a role in your research, you can insert the genes in here, and then you do the mating, and then the Galfor will induce the expression of this gene of interest. For example, in here, GFP, the gene of interest is GFP. We can take a look. We can express the GFP in particular tissue based on the promoter that we choose. And then this just information of stock centers of flies. We can order the flies, we can order the mutants, the transgenics, and everything that are already available from other researchers, and we can buy it from them. And this is the database that we can use to investigate something about the flies. And let's talk about the aging science. That's just introduction actually, but so long. I'm sorry for that. I hope you now understand about the Drosophila. So the aging is characterized. We can easily say that a progressive loss of physiological integrity, and then this leads to the loss of or impaired function and increased vulnerability to death. And there are 9 based on papers produced in 2013. There are 9 hallmarks, and particularly 3: genomic instability, telomere attrition, epigenetic, and loss of proteostasis is the primary hallmarks. So this is the one that cause damage to induce the aging. And then there are antagonist hallmarks and also integrative hallmarks. You can take a look in here. And what I want to stress is what we can learn from flies. First, it's been a long time. Drosophila researchers use flies to study aging. It's 1916. And then we can take a look that this study leads to many things, including the effect of reproduction on aging, or vice versa, how aging impairs the reproduction of species. And also, Drosophila researchers try to examine like what kind of genetic basis of the aging. And then they actually found several or many of the genes are actually play a role in the aging. So this is really multi-gene scheme. And so many pathways that has been presented are also available in mammals, including humans. For example, insulin and insulin growth factor pathway. And we know insulin also available in human. And mammalian target of rapamycin, which is also available in mammals. And aside of genetic basis, they also presented how environment can lead to the accelerated aging or can impair the health of the species. And this is based on the diet. Now we know that dietary restriction also affect aging, and then oxidative stress, conditions that caused by inflammation, etc. And then they created these lovely diagrams that say this is what we have known from Drosophila. And it will be really difficult if I explain. I will not have I will not have 20 minutes for this. So I would love to give you the slides later, but I can say that many things found in Drosophila are also conserved in humans. And then this is what makes Drosophila awesome. I'm not particularly saying Drosophila is really awesome compared to other models, but we can learn many things from Drosophila. And then the combination of these, one particular interesting thing is about insulin, which has been known so many years by physicians and pharmacists about the function. And what we can say from this conserved pathway, we can have these three beautiful statements that reduce insulin signaling pathway extends the lifespan. And this has been found in Drosophila as well as in humans. And then a high sugar diet that reduces lifespan also found to augment the insulin signaling and reduce the FOXO. And the FOXO is really related to the lifespan, which is true in Drosophila and also in human. And then one particular interesting thing is we know that when we got older, it's really, difficult to run or difficult to walk and everything. We have the muscle strength decline, and this is explained beautifully using genetic experiments by a professor from Harvard and his student in 2010. That explains that FOXO is related to this kind of impairment. And then this is related to the insulin signaling pathway as well. So the loss of the muscle is characterized by the accumulation of protein aggregates. And this leads to the difficulties in movement. And it's really a prospective target for preventing aging-related muscle loss. This was published in Cell in 2010. And one particular thing that we study in my lab and we are now trying to understand is the immunosenescence, because my basic actually immunology, but then Dr. Jeff Lano asked me to present about Drosophila, and I tried to, make it easy to understand that actually we are interested in immunosenescence. And then what we now know is the gradual depression or the decline of the immune system is actually happen, and this leads to the increase in inflammation, immune-related genes that happen during the aging. And we try to understand the experiments. We did the experiments but this is not our experiment. This is just what we are now trying to understand using our experiments. So we now know that when flies becomes older, they have much production of pro-inflammatory cytokines, and we try to assemble, these experiments using this. So we just published this year. We try to use one mutant lines that express the antimicrobial peptides. Antimicrobial peptides is an example of pro-inflammatory cytokines in mammals, and Drosophila that lacks for this kind of particular genes, PGRPLB. When we use these mutants, we observe the loss of survival. So they have declined survival. They died faster compared to the wild type. And when we just simply give aspirin, anti-inflammatory drugs, we give them aspirin, and we can see that the flies actually have longer survival. This is interesting. So we are now seeking what actually happened when flies consume the aspirin, and we are now in the experiments to molecular experiments to characterize that. But unfortunately, I cannot report now because we are now trying to prepare papers about that. Sorry, still secret. But yeah, this is a simple experiment that we can do, and we can publish in a good paper in a good journal. And then, yeah, many things we. This is the last thing about the aging in Drosophila. We use this scheme actually. We try to, for example, use the compound prospective drugs. For example, if they are not decrease the mortality or decrease the fecundity or impair the locomotion, then we can further use these drugs. But if somehow the drugs, for example, increase the mortality, then this is not ideal candidate for anti-aging, but we can use it for insecticide. This is actually we can still have the results. So it is particularly interesting, and this scheme can take us to many papers actually. Okay, now what we are doing at OFG, I'm sorry, maybe I will need 2 minutes.
All right, okay.
Possible?
I was just gonna say you have 2 more minutes, so perfect.
Good guess. Okay, thank you, Dr. Ariel. Then this is what we are doing. We have current students and some alumni, and we use mutant, transgenic, and wild type flies, and we collaborate tightly with Kanazawa, UQ, Cambridge, and Manchester for taking the flies, because we obviously cannot create the mutant in here. And then we study biological events, we do drug screening, drug repositioning, and many things others. And this is what we have tried: antibacterial activities, antiviral activities related to Wolbachia, which is really interesting. And then aging in immunity, and also candidates with immunomodulatory activities. And please kindly go to this website if you are interested to learn about fruit fly, how to use fruit fly for biomedical research. This is written in Indonesian language. This is one of our outreach for students and public spectators. And you can learn what we can learn from flies. Okay, and this is what we publish our work. And then this is our collaborators. Thank you very much for these lovely collaborators. They give us the flies for free. That's more important. Okay, and to sum up, Drosophila is potential to be used as a model organisms in the aging studies. And then in vitro and in vivo experiments are cheap and easy to do, relatively rapid results, and you can expect similar findings in human, inshallah. And knowledge is the only treasure that increases on sharing. Thank you very much, Dr. Ariel and Dr. Jefilano, for the invitation.
Thank you very much, Dr. Firzan. Dr. Firzan, actually, my real training is also in immunology.
So I am
Oh,
quite familiar in the use of Drosophila to screen a lot of immunology related, whether it innate or adaptive immunity, right? So
yes,
yeah,
yes.
it's amazing. So yeah, again, as you said, I think all the animal models, not one of them is better. Just
Yeah.
each of them have their own uses, advantages.
Complementary. Yeah.
Yes, they complement each other.
Yeah. Thank
Thank
you,
you.
Dr. Pradipta.
Thank you. Thank you very much, Dr. Fizan. Again, would like to kindly remind, if there's any question, please just write it down on the chat or keep it for later when we have our Q&A session. We are honored to welcome our next speaker, a group from the lab in Osaka University. And the first speaker will be Mr. Daniel Semi, currently the PhD candidate in Ishitani Sensei's lab in Osaka University. And he will tell about his work over there in relations to the use of killifish. Yeah, Semi-san. Yeah? And then right after that, we are honored to welcome Kotabe Sensei with a very reputable dissertation on the elucidating mechanisms for the establishment of dorsal and ventral compartment in teleosomite. And he will also enlighten us more on the wide variety of uses on killifish, especially in molecular and molecular aging research. So we will now give the time to, I think, Mr. Semi first, yeah, and then Abe Sensei.
Thank you very much. And actually, so I and Mr. Daniel will present consecutively.
Okay.
And I'll share the slide. Okay. So, okay, thank you very much for very kind introduction. And hello everyone, I'm Kotabe from Osaka University, Japan. And thank you very much for giving us a great opportunity to present here. And today we are going to introduce an emerging vertebrate model for aging study that have extremely short lifespan, turquoise killifish. And this is turquoise killifish, a very colorful small fish, and its size is about 4 centimeters long. And surprisingly, killifish ages really rapidly like this, just in several months. So now killifish is a very promising model vertebrate for aging study. However, I think most of you may not know about killifish very much. So you don't know about advantages, characteristics, and progresses of aging study using killifish. So today we'd like to introduce these points. And this presentation consists of 2 parts. And in the first part, Mr. Daniel Semi will talk general introduction. And then in the second part, I'd like to introduce some of our progresses about killifish study. So Daniel, please start.
So thank you very much, Abe-san, for the floor. So as mentioned in previous presentation, and as all of you know, so the aging process can be observed clearly from the outside phenotype. So for example, in human, we can observe when we age, the skin will go wrinkle and our hair will go gray. But aside from looks, during the aging process, an organism will also undergo a decline of function. And this decline of function will lead to several pathological problems, such as neurodegenerative diseases, metabolic disorder, and even cancer. So as shown in this graph, in the modern world, the human longevity, or the maximum lifespan of human, is gradually increasing. So while this is really good for us, because this means that we get to live longer, on the other hand, this also means that the aging population also increased significantly as well. So to balance this increase in aging population, there is a need to improve the health condition or the health span of the aging population. So therefore, understanding the mechanism that controls the aging process is very important. So as previous presenters have already mentioned, that scientists have been using the animal model as an experimental model to understand the mechanism that controls aging. So as mentioned previously, aging studies is currently dominated by non-vertebrate models, such as the previously mentioned yeast, the C. elegans, or the worms, and the flies, or the Drosophila. So from these animals, so from non-vertebrate models, there are many important processes related to aging that has been uncovered. So previously it was mentioned the IGF pathways and also the TOR pathways. However, non-vertebrate models such as yeast, worms, and flies, even though it is very great and useful models for aging, cannot fully recapitulate the process of human or vertebrate aging. So the reason is because non-vertebrate models has several disadvantages, such as lack of organs, such as blood, bones, and, well, adaptive immune system. And these organs and systems are very important in studying the human pathology, actually. So on the other hand, vertebrate models such as mouse and zebrafish has a relatively long lifespan and a higher maintenance cost. So to overcome this problem, our lab uses the African turquoise killifish, which is a vertebrate with a very short lifespan that can live in captivity. So the lifespan of killifish is only around 3 to 6 months. So it's really, really fast. So next I will explain a little bit more about the origins of killifish. So the short lifespan of killifish is probably caused by the evolutionary pressure of its original habitat. So the killifish came from the ephemeral water or transient water in the Southeast African region. So in this region, the water only forms during rainy season. And during this season, the killifish will then mature and reproduce. On the other hand, to survive the dry season, the killifish has evolved another interesting mechanism, actually, called the embryonic diapause. So during the diapause, the embryo will halt its embryonic development so that it can survive the dry season. So next, to mimic its natural habitats, we incubate the killifish embryos in coconut pits inside the petri dish, as shown here. And after around 1 month or 3 weeks, we can hatch the fish and release it to the water system. So as you can see here, after reaching sexual maturation, as Abe-san has mentioned, the killifish will show a bright color skin. The male killifish will show a bright color skin, while the female will stay translucent or colorless, as shown here. So in comparison to human, while human has an average lifespan of 80 years, the killifish has an average lifespan of 4 to 6 months. So this shows that killifish has a very high-speed aging, undergoes a very high-speed aging process. But most importantly, the aged killifish showed similar aging phenotype to those of humans, such as mitochondrial function decline, increased in tissue damage, and cognitive impairment. So besides from phenotype, so environmental intervention, such as mentioned here, dietary restriction, temperature change, and drug treatment like resveratrol, also affect the lifespan of killifish. So this is similar to the findings that is found in non-vertebrate models as well as in humans. So up until this point, I've already mentioned a lot of advantages of using the killifish. So in the next slide, I will mention more advantages actually of the killifish. So the most interesting advantages, other advantages of killifish is that there is actually 2 different strains of killifish. So the GRZ strain, or the short lifespan, so the one that is mainly being used for aging study, and also the MZRM strain. So this one has a lifespan that is actually double the GRZ strain. So what I want to say is that by comparing these 2 strains, we can further uncover the mechanism that controls the aging process. So lastly, the genome of a killifish is already sequenced and its database is readily available. And parallel to the genome database availability, combined with genomic engineering methods, we can easily establish a transgenic line using several tools, so like Toll-to transpose system and CRISPR-Cas9 system. So as a summary, a killifish is a vertebrate with a very short lifespan. So it lifespan average on 3 to 6 months, and which is very suitable for aging studies. And apart from the aging speed, the aged killifish also reflects human and other animal model aging processes. And adding to the other advantages, such as different strain, genome database, and genomic manipulation, turquoise killifish can open new possibilities to understand vertebrate aging. So that is all about the introduction of killifish. Next, for the details of our lab research, I will give the floor to Abhas.
Yeah, okay, thank you, Daniel. Okay, then in the second section, I'd like to introduce the progresses of aging study in our laboratory. So this is, actually, we started the aging study about 5 years ago, and now we established a large-scale killifish husbandry system like this. And in here, about 1,000 killifish can be maintained. And this is one of the biggest killifish husbandry system in Japan. And using this system, we've been conducting aging study of killifish. And today, I'd like to introduce 3 topics about our progress. And so killifish has relatively short history as a model animal. So experimental techniques applied for killifish is still very limited. So in order to improve this condition, now we established very rapid genetic manipulation technologies for killifish. And second, in order to further establish the basis of aging study of killifish, we further analyzed the aging phenotypes of killifish that are very well conserved across vertebrates, including human. And finally, we recently started the analysis of mechanisms of vertebrate aging using killifish. And now we are getting some interesting findings, so I'd like to introduce some of them. So let's start from the first topic. So as you know, genetic manipulation is an essential technology for model animal. However, conventional technology takes a very long time for the establishment of transgenic animal. And specifically, in the case of killifish, it takes about 7 months before starting analysis. And this is the workflow. And I don't get into detail, but as you see, so we need at least 3 generations to get homozygous transgenic. So it takes a very long time. So in order to shorten this process, now we try to apply genetic manipulation technologies in which the transgenic animal can be analyzed in the first generation, so called F0 generation. So for this, we applied F0 analysis of gene knockout fish into killifish. And we focused on multiple target CRISPR strategy. As you know, CRISPR-Cas9 system is a very useful genetic manipulation technology, and CRISPR system uses small guide RNA, sgRNA, inside the target gene. And in this strategy, not only single, but by using multiple sgRNAs, then efficiency of the gene knockout is greatly increased. This strategy is reported in mouse and zebrafish. And so now we tried to apply this strategy into killifish for the first time. And in order to verify this technology in killifish, we first targeted tyrosinase gene. this gene is producing black pigment. And this is the result of a conventional normal CRISPR-Cas9 system in F0 generation. So as you see, the tyrosinase gene is partially knocked out, but still, as you can see, some pigments are still remained. But on the other hand, surprisingly, by using not single, but the triple sgRNAs, we succeeded in the almost complete whole body knockout of this gene. And they shows this kind of complete albino phenotype. And importantly, the efficiency is also very, very high. And almost 100% of injected F0 generation shows this kind of complete albino phenotype. So by this method, we succeeded in the shortening of the transgenic generation from 7 months to about 1 month. So this is very, very powerful tool to examine the gene function in killifish aging. And also, we applied F0 analysis of knock-in-based reporter fish into killifish. And here, we used enhancer trapping system by CRISPR-Cas9 mediated knock-in. And here, I omit the detailed method, but anyway, by using this method in killifish, so we succeeded in the visualization of blood cells of killifish like this, and also the bone of killifish like this. So these fishes are actually F0 first generation. So by using this method, we can analyze the gene expression and also tissue morphology during the course of aging, even in the first F0 generation. So now, by the establishment of these technologies, we can rapidly analyze the gene functions and also the expression during the course of killifish aging in the first generation, very, very rapidly. Okay, next, let's move on to the second topic, the analysis of aging phenotypes. And so as you see in killifish, apparently aging proceeds like this. However, the identification of specific molecular and phenotypic biomarkers of aging in each tissue and organ are still insufficient. So to get more insights, we performed these two analyses. And I'll explain one by one. First is the whole body micro CT scanning. As you know, CT scanning is a technology to visualize tissue morphology by using X-ray. And actually this image is incorrect. And actual micro CT scan machine is like this. And by using this micro CT scanning, so we can obtain very high resolution tissue morphology image of very small sample. And this is the result. So as you see, we can observe very high resolution tissue morphology of killifish from head to head, trunk, and tail. And by using this method, actually we observe very typical aging phenotypes also in killifish. And this is an example. And this is a horizontal section of killifish body. And as you see, we can observe severe muscle atrophy and also the atrophy of the liver. In addition, interestingly, we further observed the brain deformation in old killifish like this. So from this result, we can clearly observe the very typical aging phenotypes, so that are very well conserved across vertebrates, including human. And next, in order to get further understanding of molecular biomarkers, we performed transcriptome analysis. So for transcriptome analysis, we collected several tissues at several time points. So from young adult to super old fish. And this is a result of the RNA sequencing. And actually, these are the summary of expression profiles. And notably, expression profiles can be classified according to their ages. So this suggests that the transcriptome data seem to reflect aging trajectory. And we further examine the specific molecular biomarkers in each tissue. And today, I'd like to present the molecular biomarker of skeletal muscle as an example. So one of a very well known conserved biomarker of vertebrate skeletal muscle aging is upregulation of ubiquitin ligases. So as you know, polyubiquitination of protein causes protein degradation. And it is very well known that in vertebrates, including human, abnormal upregulation of FBXO32 and TRIM63 cause the abnormal protein degradation, which leads to aging-related muscle atrophy. And notably, we found that the expression level of these genes are actually dramatically upregulated also in killifish muscle during the course of aging. In addition, we also found that another very well conserved biomarker, decrease of muscle regeneration ability, is also conserved in killifish. So during the muscle regeneration, these differentiation markers and muscle fiber markers are upregulated. So it is well known that the expression level is gradually decreased during the course of aging. And actually, like this, we also observed that the expression level of these regeneration markers are significantly decreased in killifish aging muscle. So these results strongly suggest that the process of skeletal muscle aging is very well conserved also in killifish. But of course, as you know, the underlying mechanisms of this muscle atrophy are still very largely unclear in any species, of course, including human. And so this means that Tacos killifish can be a very good model for the study of muscle aging molecular mechanism. So in this way, we further revealed that Tacos killifish shows very typical morphological and molecular biomarkers of aging. So this is very good model for human-related aging study. And finally, let me explain about the analysis of the mechanisms of vertebrate aging. And today, I'd like to introduce these two analyses. So first is in vivo visualization of aging-related signaling activity. So as you know, a lot of previous studies suggest that many signaling pathways are involved in the regulation of aging. However, in order to exactly understand the functions of these signaling pathways in vivo, we need to clarify when and where and how those signaling activities changes during aging in the body. So to know that, we've been making transgenic killifish in which signaling activities are visualized like this in vivo. And now we established transgenic killifish in which the signaling activity of these pathways are actually visualized in vivo. And today, as an example, I'd like to show the result of the ER stress response or transgenic killifish. So as you know, protein aggregation is the hallmark of aging, especially in neurodegeneration. And it is thought that during aging, many kinds of stresses cause unfolded protein accumulation. And this leads to the increase of endoplasmic reticulum stress, ER stress response. So ER stress response can be an indicator of protein homeostasis failure. However, especially in vertebrates, the dynamics of ER stress response in the whole body during natural aging is still very largely unclear. So to reveal that, now we established ER stress reporter killifish, and this project is conducted by Mr. Daniel Semi. And in this reporter killifish, the activation of ER stress leads to the activation of reporter GFP gene. And this is the transgenic killifish. And as you see, by applying a protein folding inhibitor, so ER stress is activated, then this transgenic shows the significant upregulation of reporter gene, so GFP expression. And using this transgenic fish, now we observe tissue-specific dynamic change of the ER stress response during aging, like this. So the top is the young tissue and the bottom is the old tissue. And as you see, now we observe the overall, so the activation of the ER stress response in each tissue. And now we are further examining the tissue-specific and cell type-specific dynamic change of ER stress response, and also the mechanisms underlying this change. So like this, so this kind of ER stress reporter fish can be a very powerful tool to understand the mechanisms of proteostasis failure during vertebrate aging. Okay, then finally, I'd like to introduce the result of metabolome analysis of killifish aging. Now we are focusing on metabolites because, as you know, metabolites are very good biomarkers. So they are very well conserved across species, and also metabolites are very close to the phenotypes. And in addition, interestingly, recently, metabolites themselves are thought to be regulators of aging-related signaling pathways, and metabolites such as NADs or methionines are a very famous example. So now, in order to further reveal the metabolic basis of aging of vertebrate, and also in order to clarify the metabolic pathways that can regulate the organismal aging, now we perform metabolomics of turquoise killifish. So first, in order to reveal the dynamics of metabolic state of killifish, we performed comparative metabolome analysis using several tissues and compared young and old. And interestingly, we found that the metabolic state is dramatically changes specifically in the liver. So then we focused on the liver. And in order to further narrow down the important metabolite for aging, we further compared the metabolome not only between young and old, but also compared the turquoise killifish different strains that has different lifespans, as Mr. Daniel explained. So actually, there are two strains, short-lived strain and long-lived strain. And by comparing these samples, then we focused on metabolites that shows the significant changes during aging, and also that shows the different dynamics between strains, so short-lived strain and long-lived strains. And now we are focusing on metabolites. And today, please pardon me for hiding the exact metabolite name. And this is still preliminary, and we'd like to present in detail in near future. And anyway, so the amount of this metabolite is significantly high in the long-lived strains, and the amount is gradually decreased during aging. So now we hypothesize that this metabolite might have anti-aging effect. So Now, in order to increase the amount of this metabolite, we knocked out the enzyme that metabolite this metabolite X. And we confirmed by this knockout, the amount of metabolite X is increased. And interestingly, in the knockout, we are observing some younger phenotypes. And like this, the knockout shows healthy darker body color and also relatively plump body. And also we are getting some molecular signals that shows this knockout is younger. So now like this, we are investigating very possible new mechanisms of aging regulation by metabolite. So this is today's summary slide. So again, turquoise killifish ages very rapidly in just several months. And they show aging phenotypes that are very well conserved across vertebrate, including human. So turquoise killifish is a great model to understand human aging. And now we are establishing some genetic manipulation techniques. And also by combination of the visualization of signaling pathway activities and also multi-omics analysis, we are now trying hard to reveal novel aging mechanisms of vertebrates. And we hope some of you might have interest in this very good model killifish. And lastly, I'd like to thank these people, especially our boss, Professor Ishtani. Thank you very much for listening. That's all for today.
Thank you. Thank you very much, Abe Sensei and Mr. Semi. That was a very enlightening presentation. Apparently, killifish is a very interesting creature that I think would really grow in their utilization in the coming years. Yeah, because a lot of things are more discovered with it recently. All right, so I think now we can open the question and answer session. Let me just check whether there's anyone in the chat that asked a question. We would like to invite anyone who has a question to write it down in the chat if you're not feeling like talking, or if you feel like interacting, is just on the camera and politely just ask. In that case, perhaps I will ask first. Is that okay? Yeah, Dr. Jeffy, I think that's fine. So I'm just a bit of a background. I myself is currently as one of the research assistant in animal research facilities in IMERI. So I am trying to develop a facility, a core facility that could accommodate different animal models. Currently, we have for cancer, we have for neurobehavior, and we have a little bit on nutrition. But we are very excited to hear about all these different animal models. Perhaps I just have a short question to Dr. Rika first. So being very proficient in using yeast, I'm sure there are different mechanisms and weaknesses that yeast cannot explain for aging in humans, for example. But I think that could be a misunderstanding. Like, I'm sure there are hints or tips that you can tell us, like how actually analysis on yeast in terms of aging genetics or aging process can actually translate to human studies.
Thank you, Dr. Ariel, for the questions. So basically, there's a drawbacks by using yeast. This is unicellular microorganisms. As I think Abe Sensei just told us that it has no bone. It has no, something like a multicellular system, a tissue connective system. So this is when we would like to understand the aging, especially in a cellular level. So we basically can easily manage to understand in terms like how the organelle would react. So basically in a cellular level, the advantages of using yeast is, in our experience, that when we have a library compound, like natural products, like here in Indonesia, we have a very high biodiversity in terms of natural resources. So like in the plants resources that have like antioxidant activity, there are so many. So before we pursue to the multicellular system for the aging research, we can use yeast as a screening. This is very rapid screening. So instead of testing 1,000 natural products towards a multicellular system, it's so much easier to test those 1,000 products or natural products in yeast, because it just has like less than a month to age. So 20 days is already aged. So this is what we did in our laboratory. So we work in a tropical biopharmaca research center where natural products is around us. So we tested number of metabolites, crude extract, active fraction, even pure compound from many plants in Indonesia, tropical plants, and we found out 1 or 2 actually active fraction as pure compound from those plants, and we further making it to higher or multicellular organisms such as mice, and just love using Avicenna's animal model. This is really sophisticated models, I think, and the way that Avicenna's laboratory working with it, with the CT scan and creating the long-lived strain, short-lived strain, making it so much easier to figuring out. And Avicenna also working with some anti-aging compound by using a metabolomic study. So we also did and find out some of the biomarker. We found amino acids, sensei, a metabolite in yeast that actually somehow probably modulate aging in yeast, especially proline, and some of the sugar, trehalose. Yeah, I don't know, but the X metabolite that you are working with, looking forward for that.
We must wait for the publication.
Looking forward for
We must wait for it.
We're keeping eye on that result, the X metabolite. I think that's it.
But that's fantastic. That means like whichever animal model we use, some of those metabolites, some of those factors can actually be cross-checked between the models. So that's great.
It's complement to each other. So there's always a drawbacks in numbers of model organisms because we resemble human with human system. So this system is not human, but just resemble.
Right.
Yeah, I think that's it.
Perhaps I would like to channel a bit to Dr. Firzan then, since he was promoting about Drosophila. Maybe you could give us some direction on what is currently recent in the use of Drosophila for aging or immunosenescence, like you said, for example. Is it autoimmune? Is it infectious disease? Or is it maybe immunomodulator? Maybe you could tell us more about that, Dr. Firzan, please.
Thank you, Dr. Ariel. Actually, I forgot to mention the limitations of Drosophila. And basically, as we have been saying a lot recently, that all animal models can complement each other and not trying to replace, but this is kind of like integrative systems that we can have many things in our hands, and maybe somehow we can relate that to humans, right? So the limitations of Drosophila is actually they don't have it doesn't have adaptive immunity, which is the core principle of Dr. Ariel's research, right? So the Drosophila doesn't have adaptive immunity. They only have innate immunity, innate immune systems, and they lacks of vasculatures. They have open blood system, which is probably some papers mention that they can use cardiac of the Drosophila, but to me it's it's really difficult to
It's highly
use.
questionable, eh? Highly
Yes,
questionable.
yes, yes. It is published maybe in PLOS ONE, but I'm not really, I'm not really confident that this is the best tools to use for cardiovascular things. But if you want to learn about the cells, I mean, you know, the cells of the cardiac, you can use it, but not in terms of the organs, right? Because they don't have the fully functional organs like humans or maybe vertebrates for the cardiovasculars. And in terms of aging, I think most of the pathways are conserved from Drosophila to mammals. So I can confidently say that this is good model as well if you want to study in terms of metazoan species. But yeah, again, this is not vertebrate. So so we need to cross-check the system using vertebrates, which comes the killifish. I really admire the killifish, Sensei. It's really good. I think this will be a really breakthrough model in the future, as Dr. Ellis said. I'm looking forward to the publication. Hopefully we can have it soon enough, right? Yeah. So yeah, that's thing. And the recent publication using Drosophila, actually, when they explain about the sensors of the DNA, I mean, like, you know, in humans, they found, I forgot the name, SIGA-STING, right? SIGA-STING sensors. And then the Drosophila researchers recently discovered these SIGA-STING sensors in Drosophila. And this was published, 2 papers in Nature, just published probably last year, probably, I forgot. But yeah, but this is what makes Drosophila, how to say, interesting, because the genetic systems is already established, right? That's the most difficult things when we deal with the vertebrates, as Dr. Abe, Associate Professor Abe mentioned. They need to establish the CRISPR-Cas9 system first when they want to do the killifish, and this already established in Drosophila. So I think we can try, for example, Drosophila first, and then we can move to killifish, for example, when we want to define some simplified pathway first, or trying to test compounds like Dr. Rika mentioned, like large compound in vivo, and then move to the vertebrates. I think that's what I can say, Dr. Ariel.
Fantastic, Dr. Rizan. Thank you. Thank you very much.
Thank you.
That opens a lot of opportunities for anyone who is interested in using Drosophila, I'm sure. Is there any questions from the audience, perhaps? I think not yet. Okay, it's okay. I have maybe just some questions for Sami-san and Abe Sensei. Being one of the first, I'm sure, being one of the first to use killifish model for aging, do you find it like you have to repeat a certain, like, for example, a certain experiment in previous models, like in non-vertebrate models, again, just to prove that, you know, that it could be translated to vertebrates? Just imagine, let's say any of us right now are actually thinking of applying for a grant to use killifish. Perhaps we can go over there and bring it over home someday. But wonder, like, what do you think, or what should we prepare? Like, how can we prove that this killifish is actually fundamental for aging research, for example? Please.
Okay, so yeah, actually, as you say, well, anyway, we also first perform the experiments to prove the conservation. So between the, also of course, yeast, Drosophila, so the same mechanisms are actually functions in the vertebrates. So many studies, so including the IGF signal and the mTOR signal, so are the function of these signal are examined in killifish, and that is also very variable. So because actually signaling pathway itself is very well conserved, and the function is also conserved in the at least cellular level. But and actually, so for example, in the case of IGF signaling, also in mice, it is already reported that the inhibition of IGF signaling is leads to the lifespan extension. So, however, the molecular mechanism, so for example, so intertissue interaction or something is not sufficiently proved. So killifish can be a model to for the detailed experiment of the, so for example, intertissue interaction or cellular level examination in, so focusing on the tissue types. That takes very long time, for example, in mouse. So anyway, so in order to shorten the experimental time, killifish is a very good model for aging, vertebrate aging. And yeah, no, or any.
Probably as shown before, like intervention with dietary restriction, for example, it shows extended lifespan. And yeah.
Is there already a lot of references on like rearing difficulties or breeding challenges? And I'm sure, I mean, your setup looks really complicated. I'm sure, was that your own innovation, or was there already a reference to build up from?
Okay, so, and also, I think not only conserve the mechanisms, but also so novel mechanisms for aging, it can be revealed using killifish. So because so as I introduced, so by the metabolomics, we found some kinds of metabolites that might be involved in the regulation of aging, and those are actually so kind of novel, not reported metabolites. So, and we can kind of perform screening experiment, so focusing on several metabolites, and this kind of experiment is so kind of tough using mice. So killifish is also a very good model to reveal the kind of novel mechanisms.
Right. Dr. Jeffy, you have a question, please.
Yes, but actually, if someone wants to ask the question, maybe someone turn on the video if you want to ask the question, please. No? Okay, so can I ask the question? So actually, I would like to ask comprehensively to all of the speakers, because I have an idea to so I'm working in the pharmacology department. So I would like to study some drug discovery for the anti-aging in several models from yeast until killifish. Maybe it will be interesting. So I would like to ask from Dr. Rika first, maybe. So Dr. Rika, is that difficult or how difficult to make some mutation in yeast? I mean, or do you have some? Have you already got some yeast with some mutation, like maybe autophagy mutation or some mutation, say something like that? Because if I have one drug candidate, for example, and then I would like to, for example, maybe the possible mechanism will be in the autophagy mechanism, then I would like to investigate some mutation in autophagy starting from the yeast. Is that possible? For example, how difficult to make some mutation in yeast?
The difficulty is not that difficult, Dr. Jeffy. So when we would like to make a gene mutation in yeast, the tools is well developed, the tools are available commercially. So basically it's just a PCR transformation, homologous recombination, and checking the colony. And but in terms of autophagy, I don't have the strain yet, but we can develop the autophagy gene deletion mutant, and the fact that the genome of the both Saccharomyces cerevisiae and Saccharomyces bombi is available in public, so we can develop the primers easily. So the difficulty is not that difficult. It's just I can say it's conventional genetic engineering. And second one, we also this one we try to work with the Parkinson disease right now. So we have a Saccharomyces cerevisiae that synthesize alpha-synuclein intracellularly. So we introduced, or we inserted alpha-synuclein gene in the genome of the we bought a plasmid inside the Saccharomyces cerevisiae. We're still working on it to find out whether this alpha-synuclein protein is misfolded or folded correctly. We're still working on it, but both we can have a gene deletion mutant, or we also have those strains that produce or synthesize heterologous protein from human. Here we work with alpha-synuclein. Yeah, I think that's it, Dr. Jeffy.
Okay, interesting. So you just need some plasmid or gene to make.
Yeah.
Oh, okay.
Yeah, and the tools is commercially available.
So what kind of tools do you?
Yeah, the plasmid that you said, the plasmid that has a antibiotic resistant modules.
Oh, okay.
Yeah. It's available.
Okay.
In my lab, we have one.
Okay. Thank you very much.
Yes, looking forward for a collaboration, I guess.
Yeah, of course. Thank you very much. Okay. Thank you very much, Dr. Rika. So maybe the next question will be in Dr. Firzan. So yeah, I think we also need the Drosophila for the aging research, of course. So I mean, for after we find some mechanism in the yeast, and then we would like to investigate the lifespan. Maybe for sure the Drosophila will be easiest way to investigate that. So my question is, so is that not difficult to maintain the Drosophila in laboratories? Is that correct? So how about the cost? Then and have you ever performed some CRISPR-Cas9 using Drosophila, maybe like Dr. Abe and also Mr. Daniels performed in the killifish, maybe? Thank you very much.
Thank you very much, Dr. Jefilano. So basically, to maintain Drosophila in Indonesia, I talk Indonesia because you're now in Indonesia, so it's easy and cheap. I don't know how much to be precise, but usually I only need 1 grant, like 1 dictygrant to maintain and to have like 8 students research, for example, like that. So one PDUPT grant.
Very practical tips. Very practical tips, Dr. Firzan. Very good.
Yes. Thank you.
Okay, so
and
yes,
yeah, about the I previously showed that we have several stock centers. So stock centers actually make the mutants or transgenic flies. If we want, we can order them to do so. Yeah. So Drosophila is really, I mean, if you want to have transgenic or mutant flies, it's really easy. We can just order from stock centers. We just tell them that we need this kind of mutation in this gene, for example, if you want, right? If not, you can just easily do EMS feeding, like ethyl metal sulfonate. So we fed the flies EMS, and then we screen for the phenotype of interest. This is for forward genetic screening, right? And for reverse genetic screening, you can just use stock centers. For your questions, if I ever do the CRISPR-Cas9, yes, I did when I did postdoc in Kanazawa University. It takes around 6 months to 1 year to establish particular CRISPR-Cas9 mutants, and we did that for the receptors of phagocytosis as well. Like, yeah, it's good. We can do it, but you know, if we do it in Indonesia, then it's expensive.
You need more than 1 grant, Dr. Firzan.
Yes. we need this help.
So you got the wild type Drosophila from
Yes.
someone labs,
Yeah.
or you just
Yes.
found in the nature?
No, I take from others lab. I mean, already
Okay.
established, because if we want to use as model systems, we need to at least have 20 generations in the lab established for the genetic background to be like, we can say this is 20 generations usually used for Drosophila if we want to establish that as an animal model. So
how
we
about the
have,
yes? Yeah, please.
go ahead.
It's okay.
Okay.
It's
Yeah.
continue.
Yeah, this is applied when we use the wild type. So we previously have this wild type from Kanazawa University. So they have the wild type, and then I took it by plane. Like, I mean, and then in Indonesia, they just have, when we arrive at the airport, they just want to see the license, like, I mean, a particular report that this is free from bacteria, virus, and everything. So we have the PCR checking first before we take it to Indonesia for particular viruses, for example, or let's say a bacteria. Actually, as far as I'm concerned, viruses for Drosophila cannot be transmitted to humans, so it's quite safe. So it is different to mosquito. The mosquito bite, but Drosophila do not bite, so they cannot transmit the viruses easily to human. Yeah, so it's easy when you bring to your country, for example.
Oh, it's relatively safe to bring to our country. So you brought the Drosophila in the
files.
In the files.
Yes.
In the larva or the major
we try to bring both, like larva stages and also larva stages at different stages, and because they have first instars, second instar, third instars. Sometimes I bought I don't care about like which instars, like as long as larvae and then adults, because during the trip, for example, you need to stay, especially during COVID-19, it's really difficult because you need to stay in the hotels, for example. It will, if you bring only adults, they will have the embryo and then they have the larva inside the files. But if you bring only the larvae, then maybe when they become adults, we don't have good facility during the trip, and then they may die. So I tend to bring 3 files for one strain. I mean, one particular line. That's what have been taught to me.
Okay.
Sorry, this is too technical. Sorry.
Okay, yeah, of course. Thank you very much. So, last but not least, maybe this is also the important question for Dr. Arvin and also Mr. Daniel about the killifish. So maybe the similar question from Dr. Firzan, how difficult to transfer the killifish? Yeah, of course, you already told that in the previous one, but I mean, how to transfer the killifish in the egg form or in this sometime, and can we keep the fish if we don't want to use it right now? Maybe we can keep it in the egg format, keep it in the, for example, in dry I don't know, dry refrigerator or something.
Yeah. So as you mentioned, transferring is not so difficult actually. So as Daniel mentioned, the killifish goes into diapause state, so in the drying conditions. So this is very stable. So we can transfer the killifish eggs in drying condition, then easily transfer to any country maybe.
So if we don't want to use it right now, I mean, can we keep the egg later maybe? Or we should use it right now after arrive in our laboratory. I mean, if we got the egg from your laboratory, for example, should we use it right now, or we can keep it in the refrigerator, or
Yeah. That depends on the duration of the drying time, diapose time. But I think you can directly hatch, and also you can keep the without hatching for several months in the room temperature. Yeah.
Okay, interesting. Yeah, thank you very much. I think it's enough. Thank you very much.
Fantastic questions and answers, all very technical, and I hope it really helped us. Unfortunately, we have come to the end of our event today, Dr. Jeffy, at 3 o'clock. I believe this past 2 hours has been very enlightening, and thank you very much, Dr. Rika, Dr. Firzan, and Abe Sensei and Sami-san for all the enlightenment. I think we are all very open-minded now about the different animal models, and that we are looking forward to different collaborations or different publications that I'm sure we will all be proud of. I shall give back to Dr. Jeffy. Thank you.
Yeah, thank you very much, Dr. Arya, for your wonderful directing this session. So yeah, it's time for us to close this session. So thank you very much, Dr. Ariel, of course, thank you very much also Dr. Rika, Dr. Firzan, and also Dr. Abe and also Mr. Daniel. So I hope we can keep in contact or maybe perform some review about some model of aging together with Dr. Ariel maybe. Yeah, in published in some journal will be interesting, and also the more further collaborative research. Okay, thank you very much. And before I close the session, so I would like to introduce the next presentation or webinar. So in August, so this time we already know about the short-lived models of aging. So in the future webinar, so we would like to present some longest living organisms in animal that will be presented by Professor Malgehes in the Birmingham Kingdom, and also Professor Arai from KU that will talk about the centenarian, the human that will live more than 100 years old. Okay, this one. So please stay tuned in our Instagram, MVA Emerit FKUI, and we will be glad to hear you again. Okay, so thank you very much, Dr. Ariel. Thank you very much, Dr. Rika, Dr. Firzan, Dr. Abe, and also Mr. Daniel. So, and also other participants. See you in the next session. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
Thank you very much.
Thank you very much.
Thank you, Dr.
Thank you very
Jeffy.
much.
Thank you,
Jeffy, Dr.
Sensei.
Ariel.
Thank you,
Thank
Dr.
you very
Firzan.
much.
Thank you, Dr. Ariel. Thank you, Sami-chan.
Yeah,
Abe Sensei, thank you very much. Saya
very much. Thank you very much.
izin live, masih.
Ya, izin. Kasih.
Makasih, Mas.
Ya, makasih.
Sukare sama des.